All right, in this video, I wanted to go over one of the most valuable tools we have at our disposal when it's time to answer genetics questions. And that tool is the Punnett Square. So we'll talk a little bit about what the Punnett Square does. We'll think about some steps so that we can always approach these Punnett Squares correctly. And then we'll look at an example problem and think about how we can use the Punnett Square to help us answer it. So, Punnett Squares. What's the point, right? The Punnett Square simulates the mating between two parents. So we're going to set this up in a way that it's going to simulate that mating. And it's going to help us predict the results. So it's going to predict the expected frequencies of progeny from those parents. So we can use a Punnett Square to tell us how often we would expect to observe certain offspring from a given set of parents. In order to complete a Punnett Square successfully, you just need to know a couple terms that we've already talked about related to genetics and follow a few simple steps. So the most important thing here is staying organized and writing down pieces of information as you go, not getting overwhelmed, and not sort of skipping over steps. Often skipping a step will confuse us, and in the end we end up writing down the wrong answer. So those are the biggest problems. And if we just stay organized and systematic, we're going to be really successful at answering genetics questions when we use these Punnett Squares. So what steps do I suggest you use? I think I'm going to suggest that you use 5 steps, and they are these. So anytime you are going to answer a genetics question, let's follow these 5 steps. And when we look at our example problem, we'll look at each of these steps one by one. So to start, we're going to choose some symbols to represent the gene's alleles. Once we know the symbols, we'll create a list of all the possible genotypes using those symbols. And then next to each genotype, we'll write down what phenotype that organism would have. Once we have that information, we're going to scour our question for information about the parents in the cross. We're going to figure out what their genotypes are. Once we know the parents' genotypes, we're going to think about what kinds of gametes they'll make. And then using that information, we're going to complete our Punnett Square, and we'll have the answer to any question we might be trying to answer. All right, so let's consider a specific example problem. And here we're going to think about pea plant height. So remember, Mendel looked at pea plants. And one of the traits that Mendel looked at was whether the plant was tall, down here in the lower left, or short. All right, so here's the question. The stem length of pea plants is inherited such that tall stems are dominant to short stems. A pea plant that is heterozygous for tall stems is crossed with a pea plant that has a short stem. What proportion of the offspring will have tall stems? Right, so we can't simply just jump to the question and try to answer it. We need to extract information from this question in a systematic way. We'll use that to fill in our Punnett Square, and then finally, at the very end, we'll be able to answer this question: what proportion of the offspring will have tall stems? All right, so what information should we look for? We're going to do that stepwise approach. So step 1, choose some symbols to represent the gene's alleles. Here we're looking at a gene that controls stem length, and the gene either makes the plant tall or short, depending on which alleles we have. There's information in the question about the alleles. We know that tall stems are dominant. So right there, I can now choose which symbols to use. Remember, we use uppercase letters to represent the dominant trait and lowercase letters to represent the recessive trait. So whatever letter I'm going to use, I'll use the uppercase to represent tall stems. In this case, I'm going to use T's, T for tall, but you could use S's for stems. You could use L's for stem length, right? Whatever letter you want to use is fine, so long as you assign the uppercase letter to the dominant allele, and a lowercase letter to the recessive allele. So again, I'm going to use T's. So uppercase T goes with the dominant tall gene, allele of the gene, and the lowercase t is associated with the short recessive allele of the gene. All right, so that's step 1, assign the symbols. Step 2, let's create a list of all the possible genotypes and their associated phenotype. So I left my symbols down here in the lower left. So again, we're going to remember that uppercase T is for tall, lowercase t is for short. So what are the possible genotypes that we can put together out of those two symbols? Well, we can have three. We could have two uppercase T's, two dominant alleles. We could have one dominant and one recessive. Or we could have two recessive alleles. And those are all the possible combinations. So if, again, we know from the question that tall is dominant, I can now answer the question, what phenotype would this individual with two dominant alleles have? Well, they would be tall. What phenotype would this individual who has one dominant allele and one recessive allele be? Well, the dominant allele always overpowers a recessive allele if they're both there. So we will see the phenotype associated with the dominant allele. That heterozygous individual is tall. And then lastly, if we have two recessive alleles, that's when we get a short plant. So I've got this information now. I know what the allele symbols are. I know what all the possible genotypes are. So now I can move to step 3 in trying to answer this question. Determine the genotypes of the parents in the cross. So we're going to go back to the question and try to mine out the information we need. So what information is there about the parents? So it says here, a pea plant that is heterozygous for tall stem is crossed with another pea plant. So this pea plant that is heterozygous, we've learned about the genotype of that parent. Heterozygous means one of each allele, one dominant and one recessive. So we know one parent is big T, little t. Good. So that heterozygous pea plant is crossed with a pea plant that has a short stem. So the other parent has a short stem. What do we know about short-stemmed plants? Well, they have to have this little t genotype. So we've just learned about the two parents. One has the heterozygous big T, little t genotype, and the other has the homozygous recessive little t genotype. So now we can move on to the next step. We know about the parents. The next step is to determine the gamete genotypes produced by each parent. So to restate what we know so far, we've already determined the cross is between this heterozygous tall plant, big T, little t, crossed to this short plant, little t. So now we're trying to figure out what gamete genotypes can each parent produce. All right, so here's the thing I would try to remember. Gametes contain one of each letter from the parent. Sometimes, in some genetics problems, we'll have more than one letter. We'll have T's and S's. When we figure out what belongs in the gamete, they contain one of each letter. In this example, we're only using the letter T, but the letter T can either come in the dominant uppercase version or the recessive lowercase version. So when we're thinking about what the gametes contain, they contain one of those T's. So for this heterozygous tall parent, the gametes either contain the uppercase T or they contain the lowercase t. For the short parent, the gametes contain either the first little t or the second little t, but 100% of the gametes will have little t in them. Right? So we're going to use that information in the next step to fill out our Punnett square. So, step 5, we're going to complete our Punnett square. So here, I've just carried over the information about our cross. Again, we've got our heterozygous tall parent, big T, little t, crossed to our short parent, little t. And I've set up this box where I'm going to fill in my Punnett square results. Okay. So to start, on the top of the box, I'm going to write down the gamete genotypes from the first parent. So remember, for the heterozygous tall parent, big T, little t, the gametes either contain the big T or they contain the little t. So I write one gamete over each column there. Now, beside the Punnett square, I'm going to write down the gamete genotypes for the second parent. This parent has the two little t alleles, so the gametes either contain the first little t or the second little t. And so I write those next to these rows. All right. The next step is just to fill in the boxes with the allele that's above that box and the one that is beside that box. So as we go here, we'll fill in big T, little t. And then in the next box over, we'll fill in little t. And then in the bottom row, we'll fill in big T, little t. And in the last box, we'll fill in little t. At this point, I'm going to add in one more piece of information. If we referred back a few slides, we made the list of all the genotypes that were possible and what phenotype they're associated with. I'm just going to write those phenotypes in the box next to the genotypes. So big T, little t, that makes a tall plant. Little t, that makes a short plant. Again, big T, little t, we know that makes tall. Little t, short. And now we're done with our Punnett square. We have simulated a mating between these two parents, and each of the boxes represents the offspring that could be reproduced by those parents. And each box represents an equal amount of the offspring. Each box represents 1/4, or 1 in 4 of the offspring. All right, so now we have to think back to what was the very original question we got asked. The original question was, what proportion of the offspring will have tall stems? So now we can just simply look at our Punnett square and say, how many of those boxes have tall stems in them? Well, 2 of the 4 boxes do. So 2 out of 4, or 50% of the offspring, will have tall stems. What's going on with the other 50% of the offspring? Well, they have short stems, right? So 2 out of the 4 boxes have short stems in them. And that's how we approach answering these genetics questions. Break it down step by step. Write down the pieces of information that you get from the question. Choose your symbol. Write down the genotypes and phenotypes. Figure out the parent genotypes. And then set up your Punnett square and find out what the offspring will be. Once you've got that, you can answer every question that you're going to get asked. So I hope that helps. As always, let me know if you have any follow-up questions, and I will talk to you again soon.